Welcome back to the deep dive. Today, we are undertaking a critical mission to analyze the single most dynamic and I think mechanically complex system in vertebrate biology, the immune system.
And we're coming at this from a very specific angle.
Right. We're not looking at this through the lens of disease or overall physiology, but really focusing on the molecular machinery that makes defense possible.
That's the core of our deep dive. We are treating the immune system as a sophisticated factory built entirely out of precise molecular components. Our focus is squarely on the foundational biochemical principles.
So things like the specific geometric fit of protein structures, the rules governing how receptors and ligands interact.
Exactly. And the intricate signal transduction pathways that move critical information across cell membranes. If you understand the biochemistry, you understand the defense.
And when you look at it that way, you realize this system has solved two challenges that seem almost impossible for a biological entity to manage.
The first one is just staggering. It's the challenge of sheer molecular diversity. I mean, how does the body build an army capable of recognizing invaders that haven't even evolved yet?
Right.
The adaptive system has to produce this enormous potential repertoire. We're talking more than 188 distinct proteins called antibodies, and a truly astronomical 1014 receptors on T cells.
10 to the 14th specific receptors. That's a scale that just defies imagination. It highlights the raw, almost infinite potential of this defense mechanism, ready to engage basically any foreign structure.
And the second challenge is discrimination. It's the ability to take that vast, diverse arsenal and teach it to distinguish accurately and, instantly between self and non-self.
Because if the system fails there,
If it fails there, the result is autoimmunity, where
Yeah.
the body directs its own powerful weaponry inward. The entire system is an evolutionary masterpiece built on variation and a ruthless selection process designed to manage that inherent risk.
okay, let's unpack the strategy this molecular army uses to manage these challenges. We have two major lines of defense, and they're differentiated by speed and specificity.
We do. We start with the rapid, ancient, innate immune system. It's found in all multicellular life, and it relies on general, conserved molecular signatures common to many invaders.
So it's an immediate, though generalized, response.
Exactly. Then we have the specialized, highly adaptable, and genetically innovative adaptive immune system. This system uses mechanisms that mirror evolution, you get genetic variation followed by selection, to generate highly tailored, specific defenses against things it's never seen before.
And today, we'll examine how modular protein construction underpins both of these spectacular defense mechanisms.
It's all about the biochemistry.
So let's start with the sentinels, the innate system. If it's ancient, fast, and generalized, how does it physically differentiate a threat from normal cellular activity using molecules alone?
It relies on recognizing patterns, and the best understood sensors in this system are the toll-like receptors, or TLRs.
And their story is a great example of biochemical serendipity, right?
It's fantastic.
Yeah.
They were first identified in Drosophila, the fruit fly, where the toll gene was initially studied for its role in embryonic development. It was only later that its fundamental importance in insect immunity was revealed.
That structural similarity across species suggests a very successful, highly conserved design. So what is the architecture that allows these TLRs to act as such effective general-purpose flags?
The structure is highly modular and repetitive, which is characteristic of robust protein evolution. The critical part is the large extracellular domain, which is built almost entirely from numerous sequential leucine-rich repeats. We call them LRRs.
Okay, so what exactly are these LRR units? How are they built and put together?
Each LRR is a small unit, usually about 20 to 30 amino acids long. And a key feature is that about 6 of those residues are hydrophobic, usually leucine, which dictates the overall fold.
And when these repeats stack up, you said in human TLRs there are between 18 and 27 of them?
Right, when they stack up, the hydrophobic residues cluster inward, which stabilizes a large parallel beta sheet structure.
So the LRR units form a sheet, but it's not a flat sheet, is it? It's got a specific shape.
No, it coils dramatically. The sheet forms a pronounced concave sort of hook-like structure, much like the internal threads of a screw. And that concave face is the crucial recognition surface.
And this whole thing is anchored to the cell.
Yep. The entire extracellular structure sits atop a single transmembrane helix, which anchors it to the cell membrane. Then, crucially, the intracellular domain, the signaling end, is not a kinase itself. It's a specialized TIR domain, designed purely for docking other signaling proteins.
So the TLR is just waiting, poised. And
Okay.
the signal it's waiting for is the pathogen-associated molecular pattern, or PAMP. Let's make sure we understand PAMPs because they are the molecular key to the innate system's immediate speed.
PAMPs are precisely what they sound like. Molecular features that are highly conserved across entire classes of pathogens but are never, or at least very rarely, found in the host.
They're essential components for the pathogen's own structure or survival, right? So the pathogen can't easily mutate them away without basically killing itself.
That's the key. So the TLR is essentially betting on the fact that bacteria and viruses share a few structural traits that are just fundamentally foreign to us.
Can you give us a couple of the most powerful examples of these PMPs and how we detect them?
Absolutely. The classic example is lipopolysaccharide, or LPS, which you might also hear called endotoxin.
Hmm.
The specific complex glycolipid is a major component of the outer cell wall of all gram-negative bacteria, like E. coli.
And the receptor for that is human TLR4.
Exactly.
The source material highlights the sheer potency of this detection. It says that administering less than 1 microgram of LPS to a human causes severe inflammation and fever, even without any live bacteria. That's a nanogram scale reaction.
It speaks to the extreme sensitivity and amplification inherent in this defense system. Another critical PMP is double-stranded RNA, dsRNA.
Which is a huge red flag for a virus.
A perfect red flag, because dsRNA is a common replication intermediate for many viruses, but it's almost absent in healthy, uninfected host cells. Human TLR3 is the receptor responsible for detecting this viral signature.
So given the highly structured nature of that LRR domain, that hook-like shape, how does it physically translate PMP recognition into an action inside the cell? How does binding generate an internal signal?
This is where the modular design of the TLR truly shines. In most instances, the LRR's concave surface doesn't bind the PMP alone. The PMP, which is often a polymeric or repeating structure itself, binds to the interface formed between two individual TLR monomers.
So if we look at TLR3, the viral dsRNA fragment needs to act as a bridge.
Exactly. The dsRNA fragment sits right in the groove, engaging recognition surfaces on two separate TLR3 molecules at the same time. This action forces the two receptor monomers to physically associate and lock together.
Which is what we call ligand-induced dimerization.
Precisely. And this principle is a foundational signaling motif we see all over biology, from growth factors to immunity.
It's a classic switch mechanism.
It is. The physical clustering of the extracellular domains pulls the two intracellular TIR domains into tight proximity. That conformational change is the switch. Once clustered, the TIR domains can efficiently recruit and activate specialized adapter proteins, which then kick off the complex downstream signal transduction cascade.
And that mechanism ensures two things: rapid speed, but also high stringency. It only fires the alarm when a relevant, often multivalent foreign structure forces the receptors to cluster.
It's a very elegant and efficient system.
Okay, so the innate system provides that rapid defense, but it's easily overwhelmed by rapidly evolving or structurally novel pathogens. So now we transition to the adaptive immune system.
The specialists.
Right. The system that solves the challenge of limitless specificity by using the molecular principles of evolution, variation, and selection to create customized defenses.
And this is the truly specialized force. It operates through two highly interconnected branches, each defined by the molecular players they use.
Let's look at the soluble side first, the humoral immune response.
The humoral response is based entirely on soluble proteins called antibodies, or immunoglobulins. These molecules function exclusively as recognition markers, binding to foreign structures and tagging them for disposal by other parts of the immune system.
And these antibodies are manufactured and secreted in huge quantities by plasma cells,
which are the differentiated, mature versions of B lymphocytes or B cells.
And the second branch is the cellular immune response, which is all about direct cell-to-cell combat.
This is mediated by T lymphocytes.
Yes.
And here we have a clear division of labor. First, the cytotoxic T lymphocytes, the killer T cells, whose job is to patrol the body and directly induce programmed death, apoptosis, in host cells that have been infected internally.
And then the crucial helper T lymphocytes.
The helper T cells,
Yeah.
they are the central orchestrators. They're responsible for receiving an alarm signal and then boosting the production and activity of both the B cells and the cytotoxic T cells. Without helper T cells, the adaptive response pretty much stalls.
Okay, before we dive into the structures, we need to clarify the language of recognition, because there are 3 terms that describe the target and they're not interchangeable.
This is really important. Let's define them precisely. The general term is antigen. That's any foreign macromolecule, usually a protein or polysaccharide, that's capable of binding selectively to an antibody or a T cell receptor.
Okay, so an antigen is anything that can be bound. But if that binding in a living physiological context is powerful enough to actually stimulate a full immune response, cell proliferation, signaling, antibody production, then we call it an immunogen.
Right, so all immunogens are antigens, but not all antigens are strong enough to be immunogens.
And finally, the smallest molecular piece of the puzzle.
That's the epitope, or the antigenic determinant. This is the tiny, specific chemical site, maybe 7 or 8 amino acids on a protein chain, or particular sugar arrangement, that the antibody or T-cell receptor physically recognizes and binds to.
And this is crucial because each B cell and each T cell is dedicated to recognizing only one single epitope.
One epitope. That specificity is absolute.
That sets the stage for the complexity ahead. We're ready to examine the structure of the humoral system's defining weapon, the antibody.
We should start with the workhorse of the humoral system, immunoglobulin G, or IgG. It's the most abundant antibody in the serum, present at high concentrations, about 12 milligrams per milliliter.
And structurally, IgG is the classic Y-shaped protein. It's defined as an L2H2T2 heterotetramer.
Meaning 2 identical light chains, L, and 2 identical larger heavy chains, H.
And these 4 chains are all held together by a combination of noncovalent interactions and critically by multiple disulfide bonds, right?
Yes, both interchain and interchain disulfide bonds, which give immense stability to that overall Y shape.
The fundamental functional architecture of the IgG molecule was elegantly revealed by the experiment Rodney Porter did back in 1959 using just basic enzymology.
It was a beautiful piece of reductionist biochemistry. Porter used limited proteolysis, a careful partial digestion of the IgG molecule with the protease papain. This gave him 3 distinct polypeptide fragments, each about 50 kilod.
And this revealed the functional segregation of the molecule. 2 of the fragments were identical and they could still bind the antigen.
Right. Those were named Fab for fragment antigen binding. These form the 2 arms of the Y shape.
And the third fragment, which had none of the antigen binding capability, was easily crystallized.
So it's named FTC for fragment, crystallizable. This fragment is the stem of the Y, and its primary function is to mediate the subsequent effector functions of the antibody.
So things like initiating the complement cascade that punches holes in foreign cells, or binding to macrophage receptors to trigger phagocytosis.
That's the FT's job.
So looking at the assembled structure, how do the chains physically contribute to these functional fragments?
The 2 Fab arms are formed by the amino-terminal domains of the heavy chains, combined with the entirety of the light chains. The FC stem is composed exclusively of the carboxyl terminal constant domains of the 2 heavy chains.
Okay, so papain cleaves the heavy chains just above the disulfide bonds that link everything together.
Exactly. It separates the Fab arms from the FTC stem.
What's remarkable about this Y shape is that it's not rigid. There's a structural feature that allows the molecule to be remarkably adaptable.
That adaptability is called segmental flexibility. The region connecting the Fab arms to the FEC stem, the hinge region, is a flexible polypeptide linker within the heavy chains.
And this flexibility isn't just for show, it's crucial to function.
Absolutely. It allows the 2 antigen binding sites to adopt an incredibly wide range of angles and distances with respect to the stem. They can swing nearly 180 degrees sometimes.
I can see why that's so important. If an antigen, like a virus particle, has repeating epitopes, that flexibility lets the 2 Fab arms adjust their spacing to match the distance between those epitopes.
Which maximizes the chance of simultaneous binding. Segmental flexibility ensures that the antibody can efficiently cross-link multiple antigens on a target surface, whether that's a bacterium or a viral coat, leading to highly stable binding.
Now, IgG is just one of 5 major immunoglobulin classes. They all share that basic 2-QH22 unit structure, but they differ drastically in their heavy chains and therefore their effector functions.
Right. The heavy chain defines the class. IgG, which we just covered, uses gamma chains and is a simple monomer with 2 binding sites. Next, we have IgM, defined by MUT chains. This one is the structural powerhouse.
IgM is the initial responder, right? The first class to appear in serum after initial exposure to an antigen. And structurally, it's typically a giant molecule.
It's usually a pentamer. That means 5 of those HELD2-2H2 units are joined together by an additional polypeptide, the J chain. This structure gives IgM a staggering 10 potential recognition sites.
This structure forces us to clarify the difference between affinity and avidity.
This is a critical distinction in biochemistry. Affinity refers to the strength of the noncovalent interaction at a single binding site.
Okay, just one-on-one strength.
Right. Avidity, on the other hand, is the overall strength of interaction that results from multiple simultaneous binding interactions occurring between the antibody and a multivalent antigen.
So even if the affinity of one of those 10 binding sites on the IgM pentamer is relatively low, the sheer fact that all 10 can potentially bind at the same time creates this massive synergistic effect.
Absolutely. The overall avidity of the IgM pentamer is extremely high, and this is essential in the early stages of infection because it allows the immune system to lock onto multivalent targets, like the repeating patterns on bacteria or viruses, with tremendous stability, even before the B cells have had time to produce high-affinity IgG.
Okay, what about IgA, the mucosal guardian?
IgA, with alpha chains, is often a dimer in its secreted form. It's the major antibody found in external secretions: saliva, tears, breast milk, mucus. Its primary role is to provide a crucial frontline defense at mucosal surfaces, neutralizing pathogens before they even get in.
And the two least understood, IgD and IgE.
IgD with delta chains is found mainly on the surface of B cells alongside IgM and plays a role in B cell activation. Its secreted function is still a bit enigmatic, though recent research points to antiparasitic roles.
And finally, IgE, the one often associated with discomfort.
IgE with epsilon chains. It's present in very low concentrations, but it mediates allergic reactions and protection against parasites. When IgE, bound to receptors on mast cells, recognizes an antigen, the resulting crosslinks trigger the mast cell to rapidly degranulate.
And that releases potent mediators like histamine.
Exactly. And that induces the familiar symptoms of smooth muscle contraction and mucus secretion. Basically, the body's attempt to violently expel the perceived threat.
We've seen that the different classes have diverse effector functions, but they all share a common building block. We need to examine how the structure of that block dictates the exquisite specificity of binding.
The key is in the differences between the relatively uniform constant regions and the hyperdiverse variable regions.
If you line up the amino acid sequences of hundreds of different antibodies, you see a clear pattern.
You do. The C-terminal half of the light chains and the last three-quarters of the heavy chains are highly conserved. These are the constant regions dictating the class. But the amino terminal domains, the variable regions, show wild, unpredictable variation in sequence. This is the site of specificity.
And the fundamental structural unit supporting both regions, constant and variable, is the immunoglobulin fold.
This is one of the most successful and widespread domains in the human proteome, found in over 750 different genes. It's an incredibly stable modular structure.
What does it look like?
It consists of two layers of antiparallel beta sheets packed tightly against each other, surrounding a central hydrophobic core. And crucially, the entire fold is stabilized by a single internal disulfide bond.
The genius of the fold is that its ends, the amino and carboxyl termini, are at opposite ends, which lets these domains be strung together linearly to form the long chains.
Exactly. Functionality, however, is introduced by specific loops that project outwards from this stable fold. And in the variable domain, there are three regions of extreme sequence variability located within these loops.
These are the hypervariable loops, better known as the complementarity-determining regions, or CDRs.
You have three CDRs in the variable domain of the light chain and three in the heavy chain. When the Fav arm assembles, the variable domains of the light and heavy chains associate intimately, bringing all six CDRs together.
So these 6 loops collectively form the single, highly specific binding surface at the tip of the antibody.
That's it. The subtle differences in the length, sequence, and conformation of these 6 loops dictate the shape and chemical properties of the binding site.
So binding an antigen is purely a biochemical event, relying on shape and chemistry, governed by the same principles as an enzyme binding its substrate.
Precisely. It is entirely driven by complementary shape and numerous non-covalent interactions. Hydrogen bonds, powerful electrostatic attractions, van der Waals forces, and hydrophobic effects. The sum of these weak forces across the entire binding interface is what creates the high specificity and high affinity.
Let's look at the physical geometry of binding, starting with a small molecule like a hapten. How does that typically interact?
Small molecules typically fit into a deep cleft or cavity formed by the convergence of the CDR loops. There's a great example of an antibody binding phosphorylcholine. X-ray crystallography showed the small molecule was perfectly nestled within a wedge-shaped cavity lined by residues from 5 of the 6 CDRs.
And what were the specific molecular handles stabilizing that fit?
The positively charged trimethylammonium group of the phosphorylcholine was buried deep inside, stabilized by electrostatic pairing with the negative charges of a glutamate and an aspartate. Meanwhile, the negatively charged phosphoryl group was held in place by a hydrogen bond to a tyrosine and an ionic bond to an arginine.
It's a perfect charge and shape match, like a custom-made molecular socket.
It is.
Mm.
Now contrast that tight pocket-based binding with a large macromolecule like a protein antigen. Does it still bind in a cavity?
It wouldn't really fit, would it?
Not usually. Large protein antigens, like the well-studied hen egg white lysozyme, typically interact across a much broader, flatter, and subtly contoured opposed surface. The contact area can be quite extensive, maybe 30 by 20 angstroms. One antibody studied bound lysozyme using all 6 CDRs.
And that increased surface area exponentially increases the binding strength.
It does. The source describes immense stabilization in that single interaction. 12 specific hydrogen bonds were formed, alongside a huge number of van der Waals contacts. The result was extraordinarily high affinity, with a dissociation constant of just 20 nanomolar.
That affinity is breathtaking. A few dozen atoms binding with the precision and strength of a covalent bond, yet still reversible.
And we also have to mention induced fit. The binding site isn't always a perfect preformed lock. Structural studies show that both the antibody and the antigen often undergo subtle conformational changes upon interaction.
So there's a bit of flexibility.
A bit of malleability, which significantly increases the repertoire of slightly different ligands that a single antibody can accommodate, further enhancing the system's effectiveness.
We come now to the central paradox of adaptive immunity. We need a repertoire of 108 to 1014 unique protein sequences, yet the entire human genome contains only about 21,000 genes. So how do you square that circle? How do you get so much variety from so little starting material?
This molecular magic relies on a groundbreaking discovery that redefined genetics in the 1970s, pioneered by scientists like Susumu Tonegawa, VDJ recombination.
What was the key finding?
The key finding was that the DNA segments encoding the variable, or V regions, and the constant, or C regions, are physically separated by thousands of base pairs in the DNA of every embryonic cell, but they're brought together and spliced during the differentiation of the lymphocyte.
So the immune system violates the classic dogma that one gene encodes one polypeptide. It uses dynamic genetic rearrangement, almost like molecular scissors and glue, to construct a new gene de novo in each developing B or T cell.
That's precisely the mechanism. Let's map out the process, beginning with the structure of the gene segments in the germline DNA. We can take the simpler light chain as our example.
Okay, so the light chain variable region is encoded by two main types of segments.
Correct. The genome contains a tandem array of approximately 40 V gene segments, each encoding roughly the first 97 residues of the variable domain, and then 5J, or joining gene segments, which encode the final 13 residues.
And these sit just upstream of the single constant gene segment.
They do. So
Yeah.
what happens during B-cell maturation is a complex of enzymes, notably the RG1 and RegG2 proteins, recognizes specific sequence signals flanking the V and J segments.
So RG acts as the molecular scissor.
It does. It randomly selects one V segment and one J segment, excises the intervening DNA, and ligates the chosen V and J segments together. This forms the complete VJ coding region. This VJ segment is then transcribed alongside the C gene, and the resulting RNA is spliced to create the mature mRNA for the light chain.
So if we have 40 V segments and 5J segments, even at this early stage, we generate $40 times 5 gets $200 different possible light chains just by this combinatorial process.
Exactly, but the heavy chain dramatically expands the complexity.
Because it requires 3 segments for its variable region.
Right. We have approximately 51 V segments, 27 D, or diversity segments, and 6 J segments. The presence of that D segment is the key structural innovation for the heavy chain.
And the rearrangement follows a 2-step assembly process.
Yes. First, a random D segment joins one of the J segments, that's DJ joining. Then, a V segment is chosen to join the newly formed DJ segment, that's VDJ joining. This forms the complete heavy chain variable region gene.
The potential for diversity starts to explode here. $51 times 27 times $6,000, that's 8,262 different heavy chains from the raw germline segments.
And if we then combine the roughly 320 total possible light chains with those 8,262 heavy chains, the total number of distinct antibodies generated by combinatorial association alone is about 2.6 million.
That's a staggering arsenal built on just over 100 gene segments. But we still need to hit $108. Where do those last 2 orders of magnitude come from? This must be where the system introduces intentional molecular randomness.
It does, through 2 crucial biochemical mechanisms that occur during the joining phase. The first is imprecise joining. The RegG1 AG2 enzymes are not perfectly precise in their cutting and realization. The cleavage often occurs at slightly variable bases near the joining points.
And the diversity is significantly amplified in the heavy chains, specifically in the third hypervariable loop, CDR3, by an enzyme that acts as a random nucleotide inserter.
That enzyme is terminal deoxyribonucleotidal transferase, or TDT. TDT is a special DNA polymerase that operates non-template directedly. It randomly inserts extra nucleotides, we call them N-nucleotides, between the V and D segments and the D and J segments.
Introducing random nucleotides at the junctions creates enormous potential for shifting the reading frame, altering the amino acid sequence, and drastically changing the physical shape of the CDR3 loop.
This imprecise joining, combined with the random insertions by TDT, multiplies the diversity generated by combinatorial association by at least 2 additional orders of magnitude, easily pushing the total functional repertoire past the 181 mark required for antibodies.
It's an incredible feat of genetic economy and calculated randomness.
It is, and even after all that genetic innovation, there's one more step of refinement that occurs after activation.
And that's somatic mutation.
Right. Once a B cell is activated by an antigen, the genes encoding the variable regions undergo a process of accelerated, localized mutation. Cells that develop mutations that increase the affinity of the antibody for the antigen are preferentially selected and allowed to proliferate.
This process, known as affinity maturation, can increase the binding affinity by up to 1,000-fold over the course of the immune response.
Leading to incredibly tight, finely tuned binding.
Okay, so now that we have a mature B cell armed with a specific high-affinity antibody, we need to know what signal is required to transform it from a passive sentinel into a full-scale antibody factory, the plasma cell.
Right. The immature B cell expresses roughly 105 identical monomeric IgM molecules on its surface, which act as its primary receptor.
But the IgM receptor, anchored to the membrane, isn't enough on its own to transmit the signal to the cell interior. It needs molecular partners.
It does. Each membrane-bound IgM is noncovalently associated with a heterodimeric membrane protein complex consisting of Ig-alpha and Ig-beta chains. And these associated chains are the key to signal transduction.
Because they contain the crucial intracellular signaling motif, the immunoreceptor tyrosine-based activation motif, or ITAM.
Exactly. The ITAM is a defined sequence, roughly 18 amino acids, characterized by specific tyrosine residues. If a tyrosine can be phosphorylated, it acts as a molecular switch.
So the ITAMs are just passive docking sites until they are phosphorylated.
Correct. And the central requirement for triggering this phosphorylation is not just antigen binding, but oligomerization or clustering of the B-cell receptor complex.
So the antigen must be multivalent, having repeating epitopes like a vast array of proteins covering a bacterial surface.
Right. The multivalent antigen acts as a physical bridge, simultaneously binding adjacent membrane-linked IgM molecules and physically forcing those entire receptor complexes to cluster together. This clustering is the master switch.
It brings the associated cytoplasmic signaling molecules into proximity, activating the protein tyrosine kinases located nearby.
Exactly. The clustering activates a member of the SIRC family of tyrosine kinases, typically Lin. Lin is now strategically positioned to phosphorylate the tyrosine residues within the ITAMs of the clustered Ig-alpha and Ig-beta chains.
So the ITAMs are now transformed. They're no longer passive. What happens next in the cascade?
Once phosphorylated, the tyrosines in the ITAMs become high-affinity docking sites for the next kinase in the pathway, Syk, the spleen tyrosine kinase.
And Syk contains 2 highly specific SH2 domains, which bind precisely onto the pair of phosphorylated tyrosines in the ITAM.
And this docking event must cause a conformational change in Syk.
It does. Binding to the phosphorylated ITAMs forces Syk into an active conformation. Now active, Syk initiates the major downstream signaling cascade. It phosphorylates numerous other signaling proteins, ultimately leading to the activation of transcription factors that stimulate B-cell growth, massive proliferation, and differentiation into antibody-secreting plasma cells.
This detailed mechanism of clustering and phosphorylation perfectly explains why small molecules, or haptens, which only have one epitope, usually fail to provoke an immune response on their own.
Because a small univalent hapten can bind to a single IgM receptor, but it can't bridge 2 receptors together.
It can induce the clustering necessary to activate Lin, phosphorylate the ITAMs, or recruit Syk. However, if that same haptens is chemically conjugated to a massive multivalent carrier protein-like keyhole limpet hemocyanin, which is over 1 million Daltons, that large structure can bridge receptors, forcing oligomerization and successfully initiating the full response.
So once the B cell is activated, it proliferates and starts producing antibodies. But the final layer of adaptation is class switching. The cell needs to change the antibody's effector function without losing its hard-won specificity.
Class switching is vital for tailoring the defense. Initially, activated B cells make secreted IgM, but through class switching, the cell can later shift its production to IgG, IgA, IgD, or IgE.
And the key is that the antigen specificity is preserved because the VDJ segment, the recognition code, remains entirely intact.
Exactly. It's mediated by a second, highly regulated gene rearrangement event. The VDJ segment, which was initially placed adjacent to the constant gene for IgM, is moved via DNA deletion and ligation to a position adjacent to a different constant gene, say Syk.
And that changes the heavy chain constant region, which dictates the antibody class and thus the function.
Switching the cell from producing a pentameric high-avidity IgM to a monomeric, highly mobile IgG, suited for high serum concentration and robust complement activation.
We must now turn our attention to the parallel challenge, defending against intracellular pathogens. These are viruses or specific bacteria that hide inside the host cell, where they are completely inaccessible to soluble antibodies.
This is the job of cell-mediated immunity patrolled by T cells.
The T cell can't just see a hidden virus inside a cell. This requires the host to employ a highly sophisticated system to display samples of its interior contents, both self and foreign, on the cell surface.
This is the major histocompatibility complex, or MHC, the molecular cut and display mechanism.
The display system for internal threats involves Class I MHC proteins, which are expressed on virtually all nucleated cells in the body. This system's job is continuous internal surveillance.
And that surveillance pathway starts in the cytoplasm. All proteins, whether they are host proteins or foreign viral proteins, are constantly being degraded by large barrel-shaped protease complexes called proteasomes.
And the resulting peptide fragments are immediately funneled into the next step.
Right. That next step requires active transport into the endoplasmic reticulum, the ER. The peptide fragments are actively pumped into the ER lumen by a specialized mechanism, the TAP protein transporter associated with antigen processing.
Which is an ABC transporter, so it uses ATP hydrolysis to move these peptides across the ER membrane.
It does. Once inside the ER, the peptides bind to the nascent Class I MHC proteins, which are awaiting assembly. This complex assembly is crucial. If the Class I MHC protein doesn't bind a peptide, it remains unstable and is ultimately degraded.
So only the MHC-I peptide complex is stable enough to be trafficked through the Golgi to the plasma membrane,
where it tenaciously grips the peptide and displays it to the outside world. If that displayed peptide is foreign, it marks the cell for induced cell suicide by cytotoxic T cells.
Let's look at the Class I structure. How is the binding groove engineered?
Class I MHC proteins, like HLA-A2, are composed of a large 44-keto alpha chain, non-covalently associated with a smaller 12-keto polypeptide called beta-2 microglobulin. The alpha-1 and alpha-2 domains of the alpha chain fold over to create the deep peptide binding groove.
And the constraint here is on the length of the peptide it can accommodate.
Class I molecules are specialized for short peptides, typically 8 to 10 residues long, which have to bind in an extended conformation. And binding stability is dictated by anchor residues.
So specific amino acid side chains within the peptide that must fit into complementary pockets within the MHC groove.
For HLA2, for example, it often requires leucine at position 2 and valine at the C-terminus. The remaining residues can vary widely, which allows a large range of peptides to be presented.
But once bound, the interaction is incredibly strong.
Remarkably so. The complex is kinetically stable. The peptide is not released for days, ensuring that the warning signal persists long enough for the patrolling T cells to successfully screen the cell surface.
Okay, so to perform this surveillance, the T cell needs a sensor capable of scanning these MHC peptide complexes.
And this is the T cell receptor, or TCR. It's a membrane-bound protein consisting of an alpha chain disulfide linked to a beta chain.
Structurally, the TCR shares the same fundamental building blocks as an antibody. It has variable and constant immunoglobulin domains, so it looks a bit like a single Fab arm, but its recognition target is exponentially more complex.
That is the crucial point of T-cell biology. The binding site formed by the variable regions recognizes the combined epitope. The TCR must recognize the foreign peptide and the MHC protein presenting it simultaneously.
So it's not just seeing the flag, it's seeing the flag and the person holding it.
That's a perfect analogy. It does not bind the peptide alone, nor the MHC protein alone. This co-recognition is the safety lock, ensuring that T cells only attack cells that are correctly displaying a threat via the host's own presentation machinery.
And just like with antibodies, the TCR requires astronomical diversity to cover the theoretical range of all possible MHC peptide combinations, so it's the same mechanism.
It's the exact same molecular mechanism discovered for antibodies, VDJ gene rearrangements. The alpha chain uses V and J segments, and the beta chain uses VD and J segments. This process, amplified by TDT and imprecise joining, yields an estimated 1014 distinct receptors.
Is the resulting structural diversity distributed the same way as in an antibody?
Not entirely. While antibodies spread their diversity across all 6 CDR loops, the genetic diversity in the TCR is concentrated overwhelmingly in the CDR3 loop of both the alpha and beta chains.
And that must be a functional advantage.
It is. The CDR3 loops are strategically positioned to interact specifically and intimately with a bound peptide sitting in the MHC groove, while the other CDR loops primarily interact with the surrounding MHC surface.
Okay, so the interaction between the TCR and the MHC peptide complex is necessary but not sufficient for activation. T cells need co-receptors to stabilize the interaction and recruit the necessary signaling machinery.
That's right. Let's start with cytotoxic T cells. They express the co-receptor CD8. This protein is a dimer that binds specifically to a conserved region, the alpha-3 domain of the Class I MHC protein.
So CD8 acts as a molecular clamp, reinforcing the relatively weak TCR-MHC interaction.
It does. So successful binding requires simultaneous engagement of the TCR, recognizing the peptide in MHC, and the CD8 co-receptor, recognizing MHCI. This tight physical clustering is what triggers the internal signaling process.
And this sounds familiar. The CD8 co-receptor cytoplasmic tail is tightly associated with the cytoplasmic tyrosine kinase, LCK.
A homologue of CAC. And when the binding event forces the whole complex to cluster, ACIC is brought into close proximity with the crucial signaling components associated with the TCR.
The TCR itself is linked to the CD3 complex, which contains the necessary ITAM sequences, just like the B-cell receptor.
Correct. ILVZ phosphorylates the ITAM sequences on the CD3 chains. These phosphorylated ITAMs immediately become docking sites for the next kinase, ZAP-70, which is the T-cell equivalent of Syk.
ZAP-70 then kicks off the full cascade, leading to a decision to kill. How does the activated cytotoxic T cell execute its primary function-inducing apoptosis?
The killer T cell docks tightly with the target cell and secretes 2 key types of proteins from internal granules. First, it secretes perforin, a 70 kilogada protein that inserts itself into the target cell membrane and polymerizes to form large pores.
Destabilizing the cell structure.
Yes, and through these pores, it delivers granzymes, which are serine proteases.
And what do the granzymes do once they're inside?
Once they enter the target cell cytoplasm, they immediately initiate the complex pathway of apoptosis, or programmed cell death. They specifically cleave and activate other cellular proteases, especially the cascases, which act in a cascade to systematically dismantle the cell, including fragmenting the cell's DNA.
Resulting in a tidy, contained death of the infected cell.
Exactly, which prevents the pathogen from bursting out and infecting its neighbors.
Okay, in contrast to the cytotoxic T cell, the helper T cell acts as a communication hub.
They express the co-receptor CD4, and they recognize peptides bound to a fundamentally different set of molecules, Class II MHC proteins.
And Class II MHC proteins are only expressed by specialized immune cells, not just any nucleated cell.
Exactly. They are restricted to professional antigen-presenting cells, or APCs, like B cells, macrophages, and dendritic cells. This functional segregation is essential for the system's logic.
And the source of the peptides presented by Class II is also segregated.
Class II MHC molecules present peptides derived from proteins that were internalized from the outside, typically via endocytosis or phagocytosis of extracellular pathogens.
This molecular segregation makes perfect sense from a systems perspective. Class I displays an internal threat, I am infected, kill me. Class II displays an external threat, I have encountered something foreign, help me coordinate the full response.
That's a perfect way to put it. Structurally, Class II is composed of 2 homologous chains, an alpha and a beta chain. The key difference in its peptide binding groove compared to Class I is that it is open at both ends.
Which allows it to accommodate significantly longer peptides, typically 13 to 18 residues long.
Correct. And the CD4 co-receptor, which has 4 immunoglobulin domains, binds specifically to the base of the Class II MHC molecule, stabilizing that weaker interaction, just like CD8 does for Class I.
So upon successful recognition, the activation cascade proceeds, but the ultimate output is entirely different.
Instead of killing the target, activated helper T cells secrete potent signaling molecules called cytokines, which include things like interleukin-2 and interferon gamma.
And these cytokines act on the neighboring APC, for instance, a B cell that has captured the same antigen, stimulating its massive growth and differentiation into plasma cells. This is the crucial link. The cellular system calls for help, and the humoral system responds with tailored antibodies.
It's a beautifully integrated system.
We must revisit the extraordinary diversity of the MHC genes themselves, known as polymorphism. It's so diverse that these molecules were first discovered because they cause the immune rejection of tissues transplanted between genetically distinct individuals.
The extent of this diversity is staggering. Humans express 6 different Class I genes and 6 different Class II genes, and there are thousands of known alleles across the population for each of these genes.
The likelihood of 2 random, unrelated people having identical MHC proteins is less than 1 in 10,000.
And this polymorphism is not random. The variation is concentrated almost entirely within the amino acid residues that line the peptide binding groove itself. This means that 2 different MHC alleles have grooves with distinct shapes and chemical preferences, leading them to bind and present entirely different sets of peptides.
Why did evolution select for such extreme diversity in a set of molecules that are so critical for basic function? Why not one standardized, highly effective presenter?
The purpose is population resilience against evolving pathogens. If the entire human population shared one single MHC allele, and a pathogen evolved a viral protein that couldn't be efficiently bound by that one MHC molecule.
That pathogen would instantly wipe out the entire species.
Exactly. By ensuring massive diversity, evolution guarantees that someone in the population will possess an allele capable of binding and presenting the critical peptides of any novel pathogen, ensuring the survival of the species.
It's a clear example of the evolutionary trade-off. Population defense over individual convenience for things like transplants.
And this molecular complexity highlights the counter-strategies employed by the most dangerous viruses, none more devastating than HIV.
The human immunodeficiency virus directly targets the orchestrators of the entire adaptive response, the helper T cells.
The virus is enveloped, expressing the glycoprotein GP120 on its surface. And this GP120 has evolved to specifically bind to the CD4 molecules on the helper T cell surface.
This binding is the initial high-affinity interaction that allows the virus to get in.
It is. It lets the associated viral protein, GP41, insert into the host membrane and initiate fusion. The consequence of successful HIV infection is the systematic destruction of the helper T cell population.
By eliminating the central call for help signal, the CD4 plus T cells, the virus cripples the ability of the B cells to differentiate and prevents the full activation of cytotoxic T cells,
leading to profound acquired immunodeficiency.
We must return to the second great challenge: discrimination. How does the immune system, with its 1014 potential specificities, maintain self-tolerance and prevent the highly reactive T and B cells from attacking the host's own tissues?
The mechanism is a selection process that takes place primarily in the thymus for T cells, where precursors called thymocytes are subjected to an incredibly stringent molecular boot camp.
And the outcome is brutal. Approximately 98% of thymocytes fail this training and undergo apoptosis.
It's a 2-step, dual selection process that determines if they are fit to serve and if they pose a threat to the self.
Step 1 is positive selection.
T cells must express a TCR that can bind with at least a weak affinity to some MHC molecule presented by the host thymic epithelial cells. If a TCR fails to interact appropriately with any MHC complex, meaning it's structurally incompetent to use the host display system, the cell dies. This ensures all surviving T cells are MHC restricted.
And step 2 is the essential guard against autoimmunity, negative selection.
Here, T cells that bind with high affinity to MHC complexes displaying self-peptides are rigorously identified. These highly self-reactive cells are either eliminated immediately via apoptosis or diverted into a regulatory T cell lineage to suppress other immune cells.
So only those T cells that bind weakly or not at all to self-peptide complexes survive and enter circulation as mature T cells.
If that stringent 98% death rate in the thymus is the cost of molecular fitness, it demonstrates the enormous risk inherent in generating such high diversity. It's a ruthless trade-off, prioritizing the safety of the host organism over resource conservation.
And when this system fails, the consequence is amplified self-destruction, which leads directly to autoimmune diseases.
These conditions, like insulin-dependent diabetes mellitus, multiple sclerosis, or rheumatoid arthritis, result when self-reactive B or T cells escape negative selection and are later amplified, leading to a targeted attack on specific host tissues.
And sometimes this failure is triggered by external events. This is the concept of molecular mimicry, right? Where a foreign antigen closely resembles a self-antigen.
Exactly. For instance, following certain streptococcus infections, the antibodies generated against the bacterial proteins sometimes cross-react with exposed self-epitopes in heart muscle tissue, leading to the severe inflammation seen in rheumatic fever.
The immune response, designed to be helpful, mistakenly targets the self due to molecular confusion.
Fortunately, the system's ability to recognize non-self-expressed proteins also offers a key benefit in health cancer surveillance.
Cancer cells often produce unique proteins that the mature immune system has never encountered.
Right, either because they are mutated versions of normal proteins, or they are proteins normally only expressed during embryonic development, like carcinoembryonic antigen or CEA.
So immune cells, recognizing these non-normally expressed antigens, are never subjected to negative selection against them in the adult.
Which means the system can mount an effective, highly specific response. These immune cells can recognize and kill tumor cells, preventing genetically damaged cells from developing into full-blown malignancies. This natural surveillance is now the basis for many modern immunotherapies.
That brings us to prevention. Vaccines. Vaccines utilize this entire intricate adaptive system to establish immunological memory safely.
Immunization exposes the host to a controlled version of the antigen, leading to the proliferation and persistence of high-affinity memory B cells and T cells. When the host encounters the real virulent pathogen later, the immune system skips the slow primary response and mounts a faster, exponentially more effective secondary response.
And we categorize vaccines based on the molecular material used for priming.
We have killed or inactivated vaccines where the pathogen is rendered non-infectious. We have live attenuated vaccines, which contain a weakened, non-virulent form of the pathogen. We use subunit vaccines, which contain only purified specific protein components. And finally, toxoid vaccines, using an inactivated form of a pathogen's toxin.
Despite the incredible success stories of vaccination, the evolutionary dynamism of pathogens remains a challenge, particularly with the development of an HIV vaccine.
The difficulty stems directly from HIV's high antigenic diversity. The virus's replication cycle is highly error-prone, resulting in a mutation rate dramatically higher than that of most other viruses. This constant, rapid mutation means the virus presents an ever-changing array of coat proteins, making it extremely difficult for the immune system to lock onto a single, stable target.
The ultimate takeaway here is that molecular structure dictates fate. The precision of VDJ rearrangement determines diversity, a flexibility of the IgG fold determines binding geometry, and the strict clustering of ITAMs determines whether a cell lives or dies.
To conclude this deep dive into molecular immunity, let's synthesize the three most important takeaways. First, the incredible sophistication of immune recognition is achieved through the modularity of proteins, defined by the ubiquitous, robust immunoglobulin fold, combined with crucial structural flexibility embedded in the 6 CDRs and the hinge region.
Second, the required vast repertoire, the 181 potential antibodies and 1014 T-cell receptors, is not pre-encoded. It is dynamically generated by the revolutionary genetic mechanism of VDJ recombination, dramatically amplified by calculated imprecision involving the terminal deoxyribonucleotidyltransferase enzyme and later refined by somatic mutation.
And finally, T cells rely on the precise, physically controlled cut-and-display system of MHC proteins to police internal versus external threats. The activation of both killer and helper T cells is absolutely contingent upon the critical signal of receptor clustering, which initiates the tightly regulated phosphorylation cascade involving key kinases like LCK and Syk and ZAP-70 acting on the ITAMs.
So here's our final thought for you to chew on. Our survival as a species is predicated on a biological gamble. The immune system generates a hyperdiverse arsenal and then ruthlessly sacrifices 98% of its T-cell precursors to ensure that the remaining 2% possess molecular fitness and self-tolerance. What does it say about the underlying biochemical complexity that evolution had to engineer such an extravagant yet necessary level of waste just to achieve perfect defense against a threat that hasn't even emerged yet?
That molecular ruthlessness is the definition of biological genius.
Indeed. Thank you for joining us for this in-depth exploration of the body's molecular defense machinery. From the entire Last Minute Lecture team, we appreciate you taking the deep dive with us. We'll see you next time.